Thank you very much. Now you hear me, right?
Yes, now we can hear
perfect.
you.
And you can see my slides.
Also we can.
Yes. Okay. So first of all, thank you very much for inviting me for this talk. It's the longest delayed talk that I ever gave. I think it's exactly like 1 year since we actually scheduled it. So I'm very glad that it actually happens. So I'm going to talk about artificial intelligence in the service of oncology.
Ewa, you must be very well prepared then.
I am. Okay. So what I will cover today, can you still hear me? Sorry?
Yes, we can. Yes.
Yeah, okay, sorry. So what I will cover today is two papers from our research lab. And the first one is the actual one that we were advertising the talk with. This is a paper published in 2019, I think. And it's about our ARA framework. I will explain it in detail in a moment. But during that year, we have also managed to produce another paper, which I also want to cover on that talk. So the first one was with a PhD student in the lab, Łukasz Rączkowski, and with Marcin Morzejko, and collaborator from pathology at the Warsaw University of Medical University, Joanna Zambonelli. And the second one was by Krzysztof Koras from our group, with collaborators from a pharma company called Merck from Germany. And I want to start by putting these two works in a common context. And this context is cancer and cancer treatment. So once the patient is either suspected to have cancer or is diagnosed with cancer, what happens is that we take a sample of that patient tumor, either by doing a biopsy or doing the surgery of the tumor. And then what happens is definitely always histopathology. So an expert pathologist investigates an image of the tumor tissue and analyzes visually how that tissue is organized. And sometimes, and this is becoming more and more common, also tumor genome analysis is performed. So, and here what I mean by this is many types of different measurements like mutations of the genes, gene expression, and other genome aberrations. And based on these two, diagnosis of the patient can be done and drugs can be prescribed. And the first paper that I want to talk about is about understanding these medical images, and the second one is about understanding the drug action on cancer cells. So about the first one, so how do we actually want machine learning or artificial intelligence to help us to understand medical images? What can ML do for us here? So first is segmentation. So we want to take that image, which looks complicated to us, and then we want to color it by the class of the tissue that is recognizable in the image. We can also then very accurately quantify these tissues that are visible in the image. How much of the tumor tissue is there? How much of immune cells, stromal cells, and other cell types? And I put these two questions in bold because this is exactly the questions that we were answering in the paper I will cover. But actually, you can also ask a lot of other super exciting questions. One of them is, how does this spatial arrangement that we see after the segmentation correlate with these mutations, which you can find in the tumor genome? Another is, how does this spatial arrangement correlate with patient survival? And these two questions we are actually currently studying. I'm not going to cover this, but we are working on a paper on this, and we found some interesting correlations. And another question is, how does the tumor microenvironment, so the immediate surrounding of the tumor, affect the tumor progression? And this is actually the center of our new project, which has been just funded, and we will be working on that. So this is actually a related slide to the topic. So I'm pretty sure you're aware that the internet is full of cats. So if you want to train a classifier, and in particular a deep learning classifier of images, which requires a lot of training data, is pretty easy, right? We know that the recent years brought this amazing breakthrough that mainly actually via the convolutional neural networks, which allow to build classifiers which are very accurate of images. In particular, they allow us to identify a cat on an image. And now if you want to say, hey, okay, this has been so amazingly successful and we want to, it should be very easy, right? Let's take this and transfer that to a medical images. Let's take the same machinery and apply it on medical images. But there we stumble on a major problem, which is the training data for medical applications is extremely hard to get. In particular, for the task that we are analyzing, there are many different types of medical images, but the stuff that we analyze is histopathological H&E stained images. Okay, they're the ones which people routinely do for cancer patients. And to give us training data, there had to be an expert pathologist who would sit in front of a computer analyzing fast images like gigabytes, each of them, taking really a lot of memory and a lot of space, right? And this person had to manually annotate regions in this visible for him or her in that image, and assign a class of the tissue to it that person only is able to recognize, okay, because it's a trained pathologist. So by hand, putting here a region and saying this is stroma and this is cancer cells and so on. And this takes a lot of time of a very well-educated person, which has a lot of other very important things to do, like diagnosing cancer patients. So to tackle that problem and actually to also, to do also several other things, we have came up with this ARA workflow. So this ARA is an acronym for 3 objectives that we put for our model. So we wanted, of course, like all other classifiers, so this is a typical objective. We wanted it to be accurate. So we wanted the predictions of the model to be as close to the truth as possible. And we also wanted it to be reliable and active. And I think these two are quite less common. And so I maybe need a little bit more explanation. So we wanted this workflow to be also reliable, meaning that it points at mislabeled training data, which is actually quite common for our application, but also happens in all other applications. You can get training images which are incorrectly labeled, and it's a very long known problem in machine learning, how to deal with such mislabeled data. We also wanted to have this is actually central to the method, to have a certainty estimation. We wanted to say which predictions that the model makes are certain and which are uncertain, because for the uncertain predictions, you can then direct an expert to verify them. And we also wanted the model to point at classes it's a classifier, a multi-class classifier. So to point at these classes that are most difficult to learn. And this particular ability allows the model also to be active. So to learn iteratively and to optimize the learning process in such a way that it is sort of happening the fastest and is using the resource of these training annotators the least. So how does this workflow look like? We start with the pathologist who takes this large slide, as I told you, and then annotates these contiguous regions and says which class they are. And then what we do is we take these and we split each contiguous region into small tiles. Okay, this allows us to standardize the data. They are all equal size, and it also multiplies the data quite a bit, but not enough, I think, still for the requirement of this CNN that we are using. So that's why we really need that kind of a loop, which allows us to learn efficiently. So we get this training data, which are these little tiles. Each of them is annotated with the same class as the region it was taken from. And then this training data is fed into our CNN or convolutional neural network based classifier. And because it's a convolutional neural network, it's accurate. Okay, let's assume that this is given. We have, of course, evaluated it is very accurate. But this is, I think, sort of the least interesting part of this entire workflow. And then once it's trained, it makes classifications, right? It makes predictions of the classes, of course, but also, so that's typical, right? It classifies. But also what it gives you is this uncertainty measure. And because it gives this uncertainty measure for every prediction, it's also reliable, okay? So in particular, it can say, okay, I am uncertain about that image. And also what it can do is go back to the pathologist and say, now in this round of training, I want you to annotate those classes that I don't understand, that I am most uncertain about, okay? Don't give me these simple tumor cells to learn. Give me more of the annoying vessels or bronchi or stromal cells, which are very difficult for me to recognize, okay? So this is why we call it active, because the model actively directs the pathologist to give it the training data. But also really cool is that it identifies candidates of images from the previous round, which the model finds mislabeled or incorrectly labeled. And it says, in this round, please correct these if they are really, check them, double check them, and if they are really mislabeled, correct them, and I will get better data in the next iteration. Okay, so this is how this entire workflow works. So now I will explain this little part, this convolutional neural network. This is the overview of the architecture. I will not dwell on the details here. We have the input and the 3 color channels. Then we have 3 main parts. They're all based on convolutions. We have the stem, the first block, and the second block. In the first block and in the second block, you also have residual connections. We have auxiliary output, which is intended to fight the so-called vanishing gradient problem. Here are all the parameters of the filter sizes, the stride sizes, and so on. But I actually don't want to go into this. What I want you to remember from that image is that we have used dropout. And dropout is important. Here, I will explain later what it means. And I will immediately go into the results. So what we have applied this workflow on is a previously published data set of colorectal cancer images We have 5,000 image patches. Each of them is 150 times 150 pixels, which is an equivalent of 74 times 74 micrometers on the image of the tissue. And these are then divided into 8 balanced classes, and these classes and representative images you can see here. This is, this is nothing like a cat, right? This is, for example, for me, I'm not trained. This is sometimes, okay, I see that they are different, but I think for me, it's still actually quite difficult it would be difficult to annotate these regions by hand myself. What are these tissue types? But this is, of course, something that the neural network, given this data, was able to learn. So we had trained the classifier to recognize cancer cells, stromal cells, complex stroma, so a mix, debris, lymphocytes, mucosal cells, adipose tissue, and empty background. Okay, so this is an accuracy, and as I told you, the model is accurate. Here on the left-hand side, actually Łukasz plotted precision versus recall. The precision is true positives divided by true positives plus false positives. Of course, we want it to be as high as possible. Recall, true positives divided by true positives plus false negatives. The reason why you see curves here is that this is drawn for a certain cutoff of the probability of assigning to each of the classes. And we see that the sort of the higher area under these curves, the better the model performs. And here, what you see in the brackets for each class is this area under this curve. The maximum is 1, and you can see this model is actually extremely well trained and extremely accurate. And we also compared, we actually made extensive predictions, which I'm not covering. If you want to see more, you can read the paper. But we also, among other things, we compared to original work by Kater and al, where they use a traditional, I think, random forest algorithm or SVM, I don't remember, but some traditional algorithm for classifying the images into classes based on some handcrafted features. And we have reduced the error, which is 1 minus accuracy, quite a lot, as you can see, almost by half, both in if we classify it into these 8 classes, and when we classified only between tumor and stromal cells. So I would say, okay, that's fantastic. It actually was quite hard to realize, let's say, obtain this. But to obtain these results, and it's fantastic work of Łukasz and of Marcin Morzejko, but Łukasz Rączkowski and Marcin Morzejko. But let's say this was expected because we are using this amazing classifier. And maybe let's go to something a little bit less common and less expected, which is this reliability aspect of the work. So I told you that we are using dropout. So first of all, what is this dropout? Dropout is a mechanism that you can use both in training and inference. So both in training the model and making predictions using the model. And this mechanism is simply turning some neurons off at random. So when it is used during training, it can be thought as a regularization. So it's a little bit disturbing this network and not allowing it to overfit to the data. I get these. Are there questions? Julia, are there questions? Or should I answer?
I think
no. Okay, because sometimes I'm getting this kind of a Zoom pop-up. But okay, if I can also
I believe there was a question, but Marcin Morzejko answered it on the chat.
Okay. Okay, so yes, perfect. If there is Marcin, Krzysztof, Łukasz, you can guys answer the questions. Yes, and we are saying, okay, there's actually, I think there's even variational in the title of the paper. I'm not sure. No, Bayesian. Okay, never mind. But some people call this dropout used during inference variational dropout. And I think this slide will explain a little bit the connection to variational inference and to Bayesian statistics. So I want now to explain you what is this, how do we actually estimate image uncertainty, okay? So to do this, I need to introduce a little bit of notation. So we have training data D, observation, which we denote O, and the class for that observation will be Y star, and Y star, it belongs to a set from 1 to M. So in our application, there were 8 classes. So we have M equal to 8. We will denote the vector of network parameters by omega. And you can associate the instance of the model with that vector of parameters. So we will denote the probability of assigning the class Y to observation O by our model that has parameters omega as P of Y star given O and omega. And you can now remember I told you there's this dropout, and you can turn off these different nodes, and each time when you do dropout, for example, during the inference, when you do dropout and you turn off different nodes, you will get different parameters. So we get omega t, the network parameters obtained for dropout t. So we exactly make this estimation of image uncertainty by exploiting this variational dropouts or the dropout during inference. So what is the sort of theoretical background behind this? So we assume here that the parameters omega are not constant values, but are random variables. And they, of course, you can consider their posterior distribution. So this posterior distribution will be the probability of omega given the data. And because we are dealing here with a complex neural network with many parameters, their joint conditional probability distribution given the data can be quite difficult. But what we are doing here, and this I think is the relation to the variational inference, is that we assume this is approximated by a simple distribution, Q. And that I'm not going to go into the details of the families of the distributions you can assume or whatever, but let's assume we have some simple distribution Q. And now this is the stuff that is very usually done in Bayesian statistics. So we will now consider the predictive distribution instead. So we will be estimating the probability of Y star, assigning class Y star to observation O given the data. Okay, and you see here in this conditional, there's no parameters anymore, right? We got rid of the omega. Why, and how do we do this? The way to go around this is to marginalize omegas. Okay, so this is done here in this equation. So you can compute the integral over the values of the network parameters, and if there there could be the joint probability of the parameters, and the if they are now random variables, we could have a joint probability of the Y star and the parameters and the omega and the observation and the data, and this now factorizes into this probability of Y star given observation and omega times the probability of omega given the data. And this is given by the model. This is the probability of assigning the class to an observation for a model. And this is this difficult distribution of omega given the data. And I told you we can replace it by a simpler distribution. So this is where this approximation sign comes in. We are replacing this P with some Q. And now there comes another approximation sign, which I'm not going to explain here because this is the harder part. This is easy. This is the harder approximation sign, which was introduced, which was proven in these papers. Yes, there are 2 papers from which we took this idea for this uncertainty approximation, namely that in the end, what they say is that you can approximate that predictive distribution, which Bayesian statisticians really like. There are no parameters here, they were marginalized. And instead of computing them in a very complicated manner, we can approximate them using dropout parameters. Okay, so we can compute 1/t times the sum of these probabilities of assigning class y star to observation given the dropout parameters. So we are now using these dropout parameters to approximate the predictive distribution, which is great. And now based on that predictive distribution, we can estimate the uncertainty. And again, this uncertainty is we will call it entropy or H, and this entropy is computed based on the entropy of this predictive distribution. So the entropy is a very well-known notion in computer science, in probability, and it's the formula for it for any probability distribution is that it's minus a sum across the values of the random variable. Here, this is the class of the observation and the probability of getting that value and the logarithm of that probability. So here is a factor of these 2. So now, and this was also this estimation measure was proposed by Gall. So we use this uncertainty of that predictive distribution for every prediction that was made by our model. And so this is okay. This is the theory behind it, and I hope some of you got how it is computed. But for those who didn't follow too much, what you should get from it is that we have some measurement of uncertainty of model predictions, which we denote H, and they are somehow based on the probabilities and some random trials of the network and how uncertain the model is about its predictions. Okay. And now how do we use it? Okay, we have some measure of uncertainty. How do we want to use it? First of all, and this is actually really cool, and this has been suggested to us by a reviewer, which is amazing. So this model can use this uncertainty to point at candidate mislabeled training images. How can it do it? We know that, we know the class in our training data, we know the classes that have been assigned by the annotator, by the pathologist. And now the model makes a prediction for the same training image, which is different than the annotated class. It makes a different prediction and the model is very certain about it. So it has a very low uncertainty. So in other words, the pathologist said, this class is this, and the model said, no, it's another class, okay? These are for us, and and the model said, no, it's another class, and I'm super certain about it. So this is a very good candidate for a mislabeled training image, right? The model thinks otherwise, and it should tell the pathologist, I think it's not what it should be. And the pathologist should verify whether the model is correct. So what we did, we actually made an experiment where we, in a controlled manner, introduced to the data wrongly annotated images. Okay, we took the same data as we had, which presumably is mostly okay, and now we put in very bad training examples. And we checked how this procedure which is, pointing at mislabeled images, how correct this procedure is. So we computed sensitivity and specificity, and sensitivity is the number of correctly pointed as mislabeled images, and to the actual number of mislabeled images, and specificity is the number of images that were correctly pointed by the model as labeled with the true class versus the number of actually labeled with the true class. And we did this, so you should look at the left-hand side. We did this for a different percentage of mislabeled images that were introduced, up to 90%, so up to like a vast majority of wrong images. And it's quite surprising how still sensitive the model is. So it's, training even on a majority of wrong images, which is like here, right? This is over 50%. Or maybe I just go through this image a little bit more. So if we have very little of mislabeled images, the model really doesn't care much about it. It learns very well what the actual truth is, and it is very well able to detect the mislabeled training images because it has a very high sensitivity and very high specificity. And with the growing number of mislabeled images, it still returns very high sensitivity, and specificity goes down, but still it's pretty amazing that it keeps these super high values. And then it also keeps a high accuracy, even though the number of mislabeled images is very large. And I think this is a super nice showcase of how good this model is in finding mislabeled images. And I want to say this is actually a very well studied problem, and a classic in machine learning, how to deal with wrongly labeled data. In particular, there's this classical paper about this. Okay, and so this is pointing at wrong images, but also another aspect of this reliability is pointing at a class which is most difficult to learn for the model. So how do we do this? So we essentially take the training images and we compute this uncertainty for them. So the model makes its predictions on them, and we just, for each class, we group the images by that class, and we check the distribution of uncertainty, which is our entropy. And you see that the model, the images belonging to stroma, and complex classes, their predictions were made by the model with the highest entropy, meaning with the highest uncertainty. And we can also see that the colors, okay, so the images which were correctly classified as complex stroma, they are in yellow here. And the ones which were misclassified here, they were actually misclassified, most of them, for lymphocytes. They actually also had the highest uncertainty. So the model, so if you do this exercise, you can just by eye see which classes are the most difficult to learn, complex and stroma, right? And also you can see, well, the model actually makes the most wrong decisions when it is also most uncertain, right? So we should really do this active learning, right? This is really a very good motivation for doing active learning. And we actually tried how well this active learning work in a somehow artificial experiment. So we took our training set, but we didn't give it entirely to our model. We were retraining the model, and we were letting the model choose which of the classes it wants to get as training data in iterations. So we were pretending we are actually doing our workflow, which I introduced on that slide. So how did it work exactly? So we initialized the training set with a small random training set, and then we were iteratively training ARA-CNN. We were assessing class uncertainties, and then we were retrieving annotations of the most uncertain classes, well, not from pathologists, but from our remaining data set, training data set. And you can see Here you actually have 3 curves. You shouldn't look at the green one because I didn't talk about it at all, but you should compare the red one with the blue one. So the red one is the entropy and the blue one is at random. So we are comparing here the learning driven by our uncertainty, driven by the model, with random training, meaning, okay, we just add these training data at random, which is actually what happens, right? We most of the time just pick the classes from the pathologist as they give us without any direction. And actually you can see that the model, the red curve, obtains the final accuracy much earlier in this process than the random curve. So we were estimating that it has sped up the learning process by 45%. So you can see already, you should appreciate how I managed to make that pathologist happy. So the pathologist is already very happy because the training process took less time. But there is more, hopefully, that we can offer with that model to make life easier for the pathologists. So first of all, the model can fully automatically make segmentation of tissue slides into classes, right? So we have this huge original image, and then we can color it into these by the different classes, and then we can very accurately quantify the class distribution, so we can count how much of this entire image is taken by that original image. By each class in that image. We can also, for each particular class, draw its probability maps and maybe more clearly show where is this class located in the tissue. Okay, so I want to summarize the first project, and I see now the time is 44 past 6, so I will only very briefly discuss the second project. So I want to summarize the first one. So it's based on a convolutional neural network model and uncertainty estimation, and it facilitates accurate predictions, reliability in the predictions, active learning, and hopefully making life easier for the pathologists. So now I want to motivate and
yes. Sorry. Yes. Maybe before you move on to the second part, we have one question from the chat. The question is regarding the ARA workflow. Was there any issue with the data sparsity in the splitting step, meaning cancer cells located only in a fragment of a picture?
So this, thank you for that question. So no, in that particular data set, we didn't have any issues because the data was previously published by a person who is actually a pathologist himself. He prepared that data for us, and we had the training data was beautifully divided into equal sized classes. Each of the 8 classes had exactly the same number of training images and so on, but this is not always the case, right? So in real life, when you and we are now analyzing actually thousands of images of lung cancer from our own collaborators and from the so-called TCGA, the Cancer something Atlas, where they have a lot of images for patients, and it's not always the case that for example, in one patient you have each of your classes well represented. But if you have so many patients, you most of the time get a lot of images, right? But there are, in these images, I could go on. There's a lot of issues. You can have, for example, really bad quality on the borders of the tissue, where the tissue was somehow ripped, and you should actually avoid it. Even the model can somehow get completely confused in these areas because the tissue doesn't look like any of the classes that you want to see there, and so on.
Okay, thank you for the answer.
Thank you. Okay, guys, so I don't want to bore everybody, but I think this is a very, very exciting project actually that we made recently. So I will just briefly make a trailer of this and And then if you guys are more interested, you can read the paper, which is on bioRxiv at the moment. Okay, so this project is about understanding drug action on cancer cell lines. So the setup, sort of the experimental setup of this project is that, again, you could, originally there was a patient sample, and then experimentalists had generated a large number of cancer so-called cell lines, where cancer cells from particular tumor type are cultured on a dish, and then they are grown, right? They're somehow I don't know how it's called, but they are nurtured and they grow in this dish now in an experiment. And then there's a database from which we took this data, and there they store experimental results where drugs were administered to these cell lines, and then the measurement was essentially assessing how well is this cancer drug killing the cells of each of different types. And at the same time, what we have is molecular feature measurements, highly dimensional data for each of the cancer cell lines. And then, a very natural machine learning task here is that we are given these drug and molecular, we are also given drug features. We can also have experiments on the drugs, and we are using them here. I didn't visualize them. But we are given drug and molecular features drug features and molecular features of a cancer cell line. And then the task is to predict the response. So how good is this, are these drugs killing the cells, which is measured by so-called AUC or IC50 values. So how good is that drug on these cell lines? And this actually is super important because this mimics the so-called precision medicine application in the clinic. The precision medicine is a very, very hot buzzword in the area we are working in. And this is actually really important because the different patients have different molecular features of their tumors, like the different cancer cell lines are very, very different between each other. And depending on what gene is mutated, what gene expression level the tumor has, the patient will eventually respond differently to the drugs. And it's amazing to have a machine learning model which can predict that. So how exactly the typical machine learning tasks can help to understand the drug action on cancer cell lines? We can do so-called feature selection. So we can check which cell line features, and there's a lot of them, so select them, which are actually predictive of the response. And this we studied with the same PhD student, Krzysztof Koras, in a recent paper in Scientific Reports, which I will not cover today. I will cover other aspects, which are multitask learning, so capturing the action of multiple drugs on multiple cancer cell lines in a single model, and interpretability or explainability. This is another buzzword, but is actually extremely important and really crucial in the medical aspect. You really want to know what is actually this model, why is this model working like it is, and which mechanisms behind the drug action on the cell lines is this model actually capturing? What has it learned? So the modeling approach that we have taken is a recommender system. So the data we had is 922 cell lines and 74 drugs, which makes 52,730 drug cell line pairs in total. So our data matrix is this AUC or IC50 values like this for these drugs and for the cell lines. And now we also have what we can call side information, which are inhibition profiling. So how strongly are the drugs inhibiting 294 kinases. Kinases are some proteins that are important for drug response in the cells. They're encoded by genes. And about these genes, we have information in the 922 cell lines. So we know how active are these kinases in the cell lines. We know the drugs inhibit certain kinases, and that's the drug information matrix. And then we also have cell line information matrix, which is what are these actually which tissue type these cell lines were derived from, but also how these different kinases are active or mutated in these cell lines, okay? And now a very, very general view of that recommender system is that you can somehow encode that site information into lower dimensional matrices and make some kind of an operation which will transform them into these data, into the response data, which is the AUC or IC50 for each drug and each cell line. And a very, very typical transformation is done by a very classical model called matrix factorization with site information, where you have linear projection of the drug and cell line features into lower dimensional vectors, and then you take a dot product to obtain your response matrix or your data matrix, okay? And what we have replaced somehow or extended this simple matrix factorization system with is our DEARS model, which is based on autoencoders for embedding and a feed-forward network for the transformation. And the DEARS stands for Drug Efficacy Estimation Recommender System. So how does it work? We have autoencoder, which is sorry if you hear my daughter. So this drug autoencoder, is trained to project non-linearly the drug information into lower dimension, 10-dimensional space, and reconstruct the drug informations in such a way we make sure that this embedding is highly informative of the drug. We have another autoencoder which projects the cell line information again into 10-dimensional space and reconstruct the cell line information. And then these two 10-dimensional vectors are combined together, not via a dot product, which is a rather simple linear operation, but again by a non-linear operator, which is a feed-forward neural network. And that's how we get our response in this model. So the model was trained on all of this data. I don't cover here how it was done, but it was done in a proper way, let's say. What I want to focus on, and I think because of time, I don't want to extend this lecture too much because it's late. But what I really want to get through here is that we have made a lot of effort in interpretability analysis. And we really wanted to make a custom approach to interpretability because the existing tools, which there are, and there's a huge field of research on explainability, explainable AI, and so on. These are very generic, like all of these gradient-based methods and so on. They don't really respond to the needs of, again, of a particular medical application or biological application. And this is what we wanted to overcome. And in particular, we, or our collaborators came up with this fantastic idea, which we implemented. And that was that we have our hidden dimensions of the cell line autoencoder, and for each dimension, I don't want to go through all of this because it's too long, but you can think of our cell line encoder and this hidden dimension here, right? This is 10-dimensional, but we have the values of each dimension for all of our 922 cell lines. And so for one hidden dimension, you can get a vector of 922 values, each value for one cell line, okay? And this vector, you can now actually correlate with whatever you want. And in particular, something that is completely outside of the model. And what we had in the model was information only about the kinases. We had, remember, around 300 kinases, but we have correlated that vector with expression of every single gene in the human genome. And that was, I mean, maybe not every single, but a lot of genes, majority of the genes. There were 17,419 genes for which expression was also measured in the same cell lines. So we also, for each gene, had a vector of 922 values of expression of that gene. And so we could correlate now our dimensions with the genes. And once we had it, we could actually, for every dimension, make a ranked list of genes where the genes are ranked by their correlation with the hidden dimension. And that was actually amazing, because based on this, we could do something that is very, very common in bioinformatics. It's not so common in machine learning, but it's very useful for interpreting the results that have something to do with gene expression. And this is GSEA, pre-ranked analysis. I will explain in a moment what that is, but that has given us for every dimension so-called enrichment in biological processes that are going on in the cell. And what is this? So this GSEA pre-ranked is actually a statistical test which computes enrichment of a set of genes, let's denote it S, on the top of a ranked list. So we took it, for each dimension now, we had a ranked list of genes that are correlated with that dimension. And then we were checking using that test, how is a certain biological process, which is actually a set of genes that do that process, how is it now, how are these genes for that process located on the top of that list? Okay, and this is what that test is doing. So it actually makes a random walk along the ranked list, and it increases the test statistics as soon as it encounters the element of that gene set. And the test statistic is actually computed as the maximum of this little mountain that you can get. So it's also, it's increasing when it encounters the element of the gene set on the ranked list, and it's decreasing when it doesn't. And the height of this little mountain is the test statistic. The p-values are computed based on permutations. But it's not, I just wanted to show you because this is actually very, very, very famous in bioinformatics, and because we used it. But the take-home message from that slide is that we were essentially checking how gene sets, which correspond to certain processes, whether they are located on the top of our ranked list based on the correlation, which actually means these genes are highly correlated with our dimension, and they all do the same thing, right? So so now what we get from this, we actually get so-called signed FDR values. This is because we don't show here p-values for that test because there were multiple tests done, so there has to be multiple testing correction made. So we are showing the red stuff is those processes that were negatively correlated with our hidden dimensions, and the blue stuff are those processes which are positively correlated. And believe me or not, but actually this interpretability analysis is extremely interesting for biologists, because when they read the names of these processes, then they get somewhat excited, right? Because they actually understand what is the DNA metabolic process, regulation of cell cycle, G2/M phase transition, and so on. And for us, it was also exciting because we have seen that actually these processes in which our dimensions are enriched, they are mutually exclusive. And that's great, because it means, it's not always the case, right? But for really majority of these processes, they tend to be enriched only in one hidden dimension. So it seems like every hidden dimension in that autoencoder has its own job, right? It's correlated with its particular set of processes. And that was actually fantastic to see. And then just one more slide of the model, and then I will have some finishing slides. So what I want to say, and I will not explain it how, but what I want to say, we ended up doing in one more step. We took it one level higher, so to say, and we have correlated now the same processes that we correlated with the hidden dimensions, which is interesting for us, right? Because we can interpret what are these hidden dimensions. They seem to be very mysterious, hidden dimension, right? But it was interesting to see what the model correlates with each dimension, but actually what is super interesting for also for our collaborators from the pharma company is now this slide, because this is the correlation of the same processes with the drugs themselves. Okay, this is actually what the model told us about the drugs which it is modeling. Okay, so we here, each of these panels corresponds to a clustering of a family of drugs. Each of them target the same pathways, signaling pathways in the cells. So they are somewhat similar to each other, you can think. And now what we are plotting are correlations of the processes with these drugs. And in brown is negative correlation, in blue is a positive correlation. And what you can see is actually pretty amazing, because there are some processes which, for example, tend to be negatively correlated with most of the drugs, regardless of their family. But there are also some processes which are actually quite specific. For example, you have these mTOR, PI3K mTOR signaling targeting drugs. And you can see that there is a relatively higher, much higher correlation of these drugs with these processes than in other families. And, you know, this actually was quite a fruitful source of discussion in our paper, biological discussion in our paper. And we really liked it as an end result. Okay, so I want to summarize project number 2 here, this DEERS model. We have proposed a deep recommender system approach to predicting response of cancer cell lines to drugs based on drug and cell line features. We propose a custom approach to model interpretability, and this has revealed, we think, general mechanisms of the drug action. So that was it about the 2 papers. And what I also want to just really briefly show in this very dense slide is that apart from the modeling sensitivity of cancer cells to drugs and modeling medical images, we actually in the group are busy doing other stuff as well. So the most of current work in the lab done by PhD students is around 2 topics, and I think the major one is tumor evolution and analyzing its microenvironment. And a number of people in the lab are working on that topic. We also have a smaller subgroup of people working on a project on generation of antimicrobial peptides. We're also working on the COVID-19 pandemic, and we more in the past, but we have done extensive work in general on analyzing high dimensional experimental medical data. So this is, I want to acknowledge here my lab members. I actually really enjoy working with all of these people here. You are seeing mostly PhD students and master students. I want to acknowledge collaborators and my funding. And finally, I want to use this opportunity. This is my last slide. I want to use this opportunity to advertise. So we have this newly funded project called Tumor Map, which is funded from Sonata BIS grant from the Polish National Science Center. We soon hopefully will start our second collaborative project funded by Merck, Germany. We have a very strong collaboration with their research and development team, and they will fund another positions. So PhD and postdoc positions are already available. There will be available more, and I really hope some of you are interested in machine learning, in applications to health, and in particular to oncology. So if you are, please apply at this email address. And yeah, with this, finally, I want to thank you for your attention, and I can take questions if anybody has questions.